Okay, so after this fairly broad overview about basic physiological principles in the morning, we now circle in on something that's of course of more relevance and perhaps also more interest to cognitive neuroscientists, namely the Bold signal proper and its properties. And the first thing that's left to do from the introduction to the Bold signal are the neural correlates, because so far we discussed essentially that, synaptic activity and restoring of membrane potentials in after action potentials, that this requires energy, and that this leads to increases in the oxygenated blood in a given region. But we haven't seen any kind of experimental evidence that tries to relate a specific form of neural activity to the Bold signal. And when I started out in cognitive neuroscience, which was around 2004, with my master, this study was quite novel. So that is work by Nikos Logothetis from 2001. It's a little bit like Michael Jackson. The question is, can you separate the artist from the work? So Nikos Logothetis, they had some trouble with monkey experiments in Tübingen over the last 5 or 10 years. So at one point they had to abandon doing monkey experiments because some animal rights activists claimed that they were not treating the monkeys well enough. It's all not that clear cut, but you might have heard about it. So that's Nikos Logothetis. This was before this. They always had trouble with animal rights activists. So this was an experiment that was done in monkeys, in anesthetized monkeys, in which in the occipital visual cortex an electrode was implanted, and hence electrical activity could be recorded, that you see here. And this was performed inside a homemade MRI scanner that they developed, so at the same time they could measure the Bold response. And so they could do simultaneous invasive recordings and MR measurements. And what they did is that they presented visual checkerboards, so like we've seen in EEG, to these monkeys, like 2 or 3 monkeys then usually, so monkey experiments usually have only very few participants. And they presented this for quite some time, so here 10 seconds. And essentially they recorded the raw electrical signal, which you see here in this black line, and the Bold signal from an area which you also see here. It's a little hard to see. That is centered around the electrode tip, so this green area. And then if you record electrical activity, like in EEG, what you typically do in invasive recordings is that you partition the data by actually looking at different essentially frequency bands in terms of filtering the data differently. And a typical thing that was done at that time was to decompose the signal into what they refer to as multiple unit activity that was or is thought to be the output of neural populations within a couple of 100 microns of the electrode tip, and local field potentials, which are thought to be synchronized dendrosomatic neural input components within millimeters of the electrode tip. So again, local field potentials is essentially the same thing that is observed also with the EEG, but at a more distant site, and everything low pass filtered by the entire skull. Multiple unit activity as the spiking output of neural populations. That's typically not observed in EEG, although some people, for example, Gabriel Curio, has been devoted like 20 years of research to also show that in EEG activity you can observe these kind of action potential outputs of EEG, but it's something that's not typical for EEG. For invasive studies, it's of course much more typical to talk about that. So essentially they decomposed the raw signal into the output of neural populations close to the electrode tip, as well as the input in terms of the synchronized dendrosomatic neural voltage fluctuations. And the question was, what of these two signals correlates more with the bold signal? And the main conclusion that they draw from this is now not based on some single trial correlations between how high was the LFP and how high was the bold, or how high was the multiple unit activity and how high was the bold, but more on the temporal profile that they observed. So they used different stimulation setups, so 24-second stimulation, 12-second stimulation, and 4-second stimulation. And what they observed is essentially that if you look here, the stimulation is on from 10 to 34 seconds, sorry, not milliseconds, seconds. They observed that the bold signal is starting to go up after some time, and then it's basically shown this plateau response with a little bit of decrease here, and the multiple unit activity, as well as the spike density function, which is a different way of looking at the raw data, but also relates to the spiking activity. They only peak here in the beginning, so output is happening from this region essentially in the beginning and then goes back to baseline. The only thing that keeps on being present in the electrophysiological signal is the LFP activity, and so essentially a more lower frequency part of the electrophysiological recording. And essentially the same observations they made for 12-second stimulation. So as you can see here, a peak of multiple unit activity and spikes in the beginning, and then going back to baseline, and the only thing being still active being the local field potentials. And based on this, so this prolonged presence of local field potentials, they reasoned that the main contributor to the bold signal might be the local field potentials rather than spiking activity. That's the entire chain of reasoning, so there's not more behind this. It's just that local field potentials stay active and stay non-zero, while also the bold signal is active. And this has led to kind of the overall belief that bold activity reflects, it's louder than before, that bold signals reflect the input processing of a region rather than the spiking output. And in a way, this is of course similar to what the EEG also reflects, which also reflects these integrative postsynaptic potentials. This was then also looked at again later, the same data with let's say a more conventional analysis, in the sense that they filtered the raw data into alpha, beta, gamma, and multiple unit activity, so making that in a way a little bit more comparable to EEG analysis. And there the finding is that the higher correlation between these filter bands of the local field potentials is with gamma activity, And so this measures the molecular information, and less so with alpha activity, so 10 Hertz. If one looks more into the EEG Fmri literature, so this is not shown here on the slide, there is the general finding that gamma and bold activity are positively correlated, while alpha activity and bold are more negatively correlated. Are they outside or in the other room? Outside, so. Good, yeah, so that's this data set that was acquired in Tübingen in the late 90s and early 2000, and basically provides still the story about bold being more related to neuronal input processing of an area than to output. In fact, I think if you read Fmri papers these days which deal with some cognitive processes, some representation of whatever task states or whatever, nobody ever discusses that. So it's not something, although you might think so, that everybody who's using the bold signal all the time, then when claims are made with respect to the function of the brain, that everybody would say, this is not the input, so this would mean this and this. It's not happening. So in the literature that you read today, the bold signal is essentially just taken as a measure of neural activity, and what kind of neural activity is then not further specified. There are actually data analytical frameworks that would allow you to dissociate these things. So you would need biophysical models that you constrain both on electrophysiological, so for example EEG data, and Fmri data, but that's also not happening that much. It's interesting research, but not happening that much. And if you look at Fmri studies, they usually don't discuss that this is not the input to this area. But this is very famous, and one should, so especially this, this is very famous, so one should know it. It's important. Good, so these are the neural correlates of the bold signal. Are there questions about the neural correlates or anything that you have encountered previously that doesn't match this or something? So these kind of investigations can be done in vitro, monkey, Fmri, and at the same time neurophysiology is happening at very few locations in the world, and less due to the ethics constraints, which also makes sense, of course. So it's not, so there's definitely much more using the bold signal as a surrogate measure of neural activity, and then using findings from bold signal to talk about neural architectures, then research on trying to elucidate which specific aspect of neural activity contributes how much to the bold signal. So
as far as I know, some countries like Switzerland is trying to prohibit monkey studies, and I don't know whether it's common in neuroscience right now, monkey studies, like in the old days. Is it common? Monkey studies.
They still exist. So in Germany, there are still a couple of labs that do monkey research, but definitely it's not as prevalent as it was. And it's, of course, always also discussed whether it's a good thing or not. And personally, I think for cognitive neuroscience, it's not really required, because if you study it, you study the monkey. If you're interested in the physiological coupling processes, I think more and more of this research is actually done in rodents, which, it's an ethical question whether you want to destroy more rodents or more monkeys or more humans. It's hard to tell. Good, so this thing is stuck. So now we basically covered this third unit, starting with an introduction to the bold signal and its more general context, so the metabolism and the hemodynamics. And the next part is then looking closer at the bolt signal, trying to understand its spatial and temporal properties, and also some of its aspects that then relate to actually data analysis. And that's what's following now. So first, it makes sense to think about the spatial resolution that can be achieved when using bolt signal for brain mapping the temporal resolution. An important thing in terms of basic general linear model analysis are the question of whether the bolt signal is linear in terms of its input function. It's important to discuss different signal to noise ratios because they exist and people mention them, and it makes sense to know what they mean, and also to know a little bit about sources of noise in Fmri. And in the very end, we briefly discussed some aspects about field strength, but that's really short. So spatial resolution. So of course, the common rhetoric on EEG versus Fmri is that EEG has great temporal resolution but poor spatial resolution, and Fmri has great spatial resolution but poor temporal resolution. So that's that's kind of the basic thing. But of course, if one looks closer, things are a little bit more complex, and certain aspects, for example, in the EEG, of course, also have very good spatial resolution. If you think about localizing early sensory dipoles, that works really well. And then there are other aspects in terms of Fmri that argue for some good temporal resolution, maybe even a little bit too good of temporal resolution in some recent publications for my taste. So it's definitely more complex than EEG is good in time and Fmri good in space. So it makes sense to look a little bit closer. So in terms of the spatial scale in the brain, this is kind of an overview so that you get a feeling. So of course at the smallest spatial scale we have things like ion channels and synapses. The cortical columns that Rainer Göbel mentioned last week. I will refer to Rainer Göbel the entire year. I always say Rainer Göbel mentioned that a year ago. The cortical columns that he mentioned in the order of millimeters, and then more the orders of where typical Fmri results reside, namely at the level of gyrals. So, for example, you find a activation blob in frontal gyrus that is then more on the order of centimeters, and the Brodmann areas that we saw, I think in the beginning here, they are on the order of square centimeters, and obviously the brain is more on the order of like 10 centimeters. So I think it's like 15 centimeters in length. That's kind of a typical thing for humans. When talking about spatial resolution and where we can actually image these kind of things, it's of course an important question of what the spatial resolution of the imaging technique is, and this of course refers to the voxel size, where the voxel size, as we learned yesterday, actually refers to results from the reconstruction of your Fourier transformed data, and the higher, or if you acquire data from higher and higher spatial frequencies, then of course your voxels can get smaller and smaller, and so it depends on your sequence and the data that you measure in k-space, how large your resulting voxel sizes actually are. Which you of course then, once you have or let's say the effective voxel sizes, because you can of course resample all of this, and even if you have no data, partition data of a voxel that shows just the same signal into two voxels, but the spatial resolution in terms of the image and the voxel sizes before any kind of resampling depend of course on the frequency stuff that you acquire. So in terms of the image, voxel size, now I'm not talking about the imaging process, but just talking about the image, depends on how big your matrix is, so how many numbers you acquire per unit space, how big your field of view is, and of course how, if you do 2D Fourier imaging, as we discussed yesterday, how thick a slice is. So if you excite at fairly distant sites, then of course you get thicker slices, and hence your voxels in one dimension and the Z dimension get larger. So the general rule is that if you want smaller voxel sizes, you need more high frequency components and hence longer volume acquisition. So if you remember our data points that we acquired there yesterday, going up to a certain spatial frequency, if you want to go to a higher spatial frequency, you of course need to acquire more data points, and then you get more. So in a way, it's tricky to increase or it's not tricky, but it's there are some trade-offs in increasing the voxel resolution, because you lose temporal resolution. On the other hand, if you use large voxels, then you induce what is known as larger partial volume effects, and that essentially means that if you have different tissue types that in one area of space, so in this case some cerebrospinal fluid, some cortex, and some white matter, that if you have a large voxel, you get one gray value from that you allocate to this region in space, and this may refer to a mixed signal from different tissue types. So partial volume meaning that one voxel is partially populated by different kinds of tissues, and this is of course something that you want to minimize if you want to make a precise claim of where in the brain something happens. So it's just a term that comes up. This is kind of the imaging side, so that you can have higher resolution in terms of your imaging process and lower resolution in terms of your imaging process. But what is as important as actually these physical aspects of creating the image or recording the data are the functional aspects. So this is an experiment where people stimulated a cortex at a given location with an electrode, and then measured the extension or the dilatation of an arteriole at some distance from that. And what these numbers here show is that are the diameters of these arterioles and how much they change after inducing electrical activity here. And as you can see, the further you go upstream, the larger the diameters. So the vessels are bigger because they branch out, obviously. But the less their relative dilatation. So here, fairly close to the stimulation site, you have dilatation of around 23%, but still you also have at some distance you have a dilatation of 10%. That means here you stimulate, but here you still see the effect of dilatation. That means that not only exactly where the electrode has induced electrical activity, you have an upregulation of blood flow, but also further upstream. So this is the thing that it's nice if you have now very small voxels that can cover only this. But if this entire patch here on the millimeter scale is actually more strongly perfused, if there's some activity somewhere, then of course these very small voxels don't really help you. Yeah? So it's always an interaction between what kind of biological effective spatial resolution you have versus your imaging process. And again, there is no clear-cut rule that holds for everything. That you can basically say, for whatever I'm interested in, the spatial resolution is 1 millimeter by 1 millimeter by 1 millimeter or something like this. But it depends. And it depends on both the cognitive process that you induce, the neurovascular coupling, the analysis, and the imaging parameters, and of course these biological factors that are happening at the level of the brain. So the question is then how far can one push these things? So the typical fMRI results that you see, as I said, if you do a basic GLM analysis, you see blobs that are at the level of gyri, so in the order of a couple of square centimeters, and they are then active, and the other things are not that active in contrast, but one can push this. So here, this shows an example from the group in Wisconsin that have contributed quite a lot to MR imaging development, and they looked at ocular dominance columns. These are areas in the visual cortex that are primarily active for one or the other eye, and with high resolution sequences focusing on the visual cortex, and extensive care that the participants here don't move, because if you move, of course, then any interesting spatial resolution will also be destroyed. They were able to show relatively stable ocular dominance column patterns here in, I guess that's right, visual cortex. So you see that some blue parts, they get the input from the left eye, and some more reddish orange parts get the input from the right eye. So this ocular dominance here. The claim or the finding is that if you look at the between session data from one participant, in the raw data, well, it's kind of similar. In the process data, it's even more similar. Looks very similar. There has been recently, so this is from 2018, there has been more work, and there's now also an interest in, so this was more on the columnar level. Currently there's some interest in the layer-specific imaging. So I'm trying to basically allocate increases and decreases in both signal, not only to the entire cortex in a given area, but to specific layers. And if you remember what we did when we discussed layers in EEG, because of their connectivity profiles, they also might support different functions like input processing or some internal processing of a cortical module and output processing. So there is some interest in terms of layer specificity, and this is some work which I'm not quite sure. Can somebody Google that, whether this actually has come out yet? So this was on bioRxiv last year, and it's a typical like Nature or something like paper, but I'm not sure it has actually been published. So it might be still under review, and reviewers still asking them to do more experiments or something. So what they did, they wanted to look at the layer-dependent activity in prefrontal cortex as a function of a cognitive task, and they had a working memory condition where people were asked to either remember a given visual stimulus or to manipulate the working memory content in terms of, I think the idea was to, what was it here? let me check what the task was. I just forgot. It was more than just remembering the stimulus. More than just remembering the stimulus, but so they had to rearrange the letter in alphabetical order. So that's that alphabetize task. So they were shown a stimulus, then there was a delay period where they didn't show the stimulus, and they had to in their head alphabetize them, so manipulate them, manipulate the content or remember it. And they also had control conditions here that where they actually controlled for the response aspect. Now, what they were interested in doing and what they what their hypothesis was, kind of an intuitive setting, was that the recurrent connections in superficial layers would be involved in the delay period when things have to be alphabetized, whereas the more deeper layers, so the output layers that in frontal cortex send motor signals, would be mainly involved in the response scenario. So this is of course a little bit hand wavy. So yeah, recurrent connections are important for manipulating memory content. Why not? Deeper layers for motor output. Yeah, I guess so. So that was their kind of hypothesis. And what they then did in order to see that in the bold responses, that for each participant they selected a specific region of interest, and then of course first had to differentiate superficial from deeper layers in their participants, and they used a dip in terms of, if I remember correctly, the myelination as a marker for, or as a border between the upper and the lower layers. So what you see here, I make it a little bigger. Is functional data, and you see that you basically have to allocate a couple of voxels of cortical depth to them being superficial and the other one to being deep layers. Then if they looked at the signal changes from these areas, what they see in the superficial layers in terms of the bold signal is that, as expected, surprisingly, during manipulation of the working memory content, the superficial layers show more bold signal during alphabetizing, as do the deeper layers to some degree. The deeper layers showing mainly activity during the action. It got a little bit more specific if they not only looked at bold, so this was only from the bold, but if they also looked at the cerebral blood volume, so what they refer to as their vasal sequence, that looks not only whether there's a change in T2 star time constant, but also it changes in the cerebral blood volume. Then it got more specific in the sense that they see during the delay mainly an increase in activity during working memory content manipulation. And again, so this is not happening here. And again, in the deep layers, they see primarily a covariation with the motor response. So this is kind of where we are currently at in terms of spatial functional resolution. So now with an interest in layer-specific MRI. There is some talk about this layer-specific MRI. There is also some modeling, so some proposals of how these both responses are generated in different layers differently. Yeah, the experimental evidence is slowly accumulating, let's say. Has anyone checked whether anyone went to bioRxiv to see whether this is published?
Yeah, it was published in this year of October.
And in which journal?
In Nature Neuroscience.
Oh, that's why I guess. So it's this year in October, so it's now essentially.
Yeah.
It's just came out. Yeah. That was kind of expected. Yeah, so this is actually where we at right now, not a year ago. Okay, so that was on spatial resolution and current thing. But yeah, again, I mean, even if this is right now and current and so on, remember they partitioned the cortex into 2 layers essentially, yeah, and they looked at the upper and the lower. So it's not like layer 1 does this, layer 2 does this, and so on. It's like superficial and deep. Good. Then temporal resolution. So it's fairly similar in terms of the story. So one thing is that temporal resolution is of course determined by the sampling rate, and the sampling rate in Fmri being called or being depending on the repetition time. So the question. If you, so a typical imaging sequence using 1 excitation per slice, when you shoot that or when you make the next excitation, and the time between 2 excitation pulses being the repetition time or TR, and if you decrease this time between excitation pulses, then of course you create higher resolution temporal resolution images, or you create more images per unit time, that's more correct. Whereas if you have long TRs, like 3 seconds, obviously you have lower temporal resolution. And this can, for example, here, I think the point is that things like the initial dip you might not see if you have this relatively low temporal resolution, while if you go from temporal resolution with a TR of 750 milliseconds to even higher temporal resolution in terms of the characteristic features of the bolt signal, you don't see any other changes, so it might not even matter. The one question that always comes in terms of temporal resolution is whether one should synchronize a stimulus and the TR, so that whenever you show a stimulus at time 0, that you then acquire data synchronized with that, so you send an excitation pulse at the same time that you send your stimulus, and then you send your next excitation pulse, or whether you want to jitter that, so that sometimes you send the stimulus with your excitation pulse, and sometimes you send your stimulus briefly after your excitation pulse, and so on. So you jitter around that, and that essentially results in 2 different kind of data patterns. If you synchronize things, then of course for each of these time points that you actually measure, you have more repeats and have kind of more reliable signals. So because here in this case you would, for example, twice measure at this time point and twice measure at this time point, and then if you average over this, then you have a good idea. If you jitter, then that means that effectively your sampling rate is increased, because you sometimes turn on the stimulus in between excitations and sometimes directly with the excitation, so you increase your effective sampling rate. On the other hand, if you use exactly the same number of trials and same imaging time, then you have for each of these data points, you have fewer repeats. So in this case, half of the repeats, because instead of measuring at the same time after stimulus onset. You twice, you measure once a little bit after stimulus onset and once a little bit more after stimulus onset. So again, it's a trade-off. So do you want reliable signals for specific time points, but then fewer time points, or do you want less reliable data for more time points? So that's something that is sometimes discussed. There is also some interest in the temporal resolution of fMRI for cognitive processes. So here, what this is just supposed to show you is that if you have a stimulus, then it's the relevant areas in the brain become active quite quickly. So you remember that we had this primary visual cortex response in the EEG at around 70 milliseconds, then some extrastriate areas, the P1 at 100 milliseconds. And in this case, if you have to do a decision, then of course also motor cortex needs to become active and you need to make a decision. That means that the interesting processes that happen in this kind of fast decision-making experiments happen in half a second. And your TR in fMRI is commonly longer than 500 milliseconds, so more on the order of 1 to 2 seconds. However, and also the bolt response, of course, has its peak only 4 seconds after such a process. However, the important thing is not so much whether there's a delay, but whether this delay and how you pick it up is reproducible and not uncertain. So this shows an example that tries to resolve temporal aspects of cognition with fMRI. So here people had to do visually guided motor responses, and what then was looked at was the differences in bold peak latencies, and this was looked at in relation to the reaction time. So the point here being that I have to be careful. So the point being that a difference in the onset of the bolt activity between the supplementary motor area and area V1 correlated with differences in reaction time, basically saying that although these differences of course occur a couple of seconds after stimulus, these differences were nevertheless positively correlated with reaction times. So that on a given trial you sometimes take longer. You only take longer, 100 milliseconds. But what also happens there is that the onset of V1 and the onset of supplementary motor area are also shifted by roughly 100 milliseconds. Whereas, and this was the point here, the difference in terms of the onset of the bolt signal between M1 and the supplemental motor area, where things are thought to be more hard-coded. So the idea is between V1 and premotor area, there are all these cognitive processes that essentially lead to your reaction time. Whereas if the signal is in the supplementary motor area and then goes to M1, that's fairly hard-coded and doesn't have to do with cognition. And there you don't see this covariation with reaction time. So you still see so you basically always have the kind of same onset difference in terms of 50 milliseconds regardless of the reaction time. So this is from 1999, hence a claim that that bolt signal latencies, although they are quite far shifted from the stimulus, but their relative latencies then still correlate with, for example, reaction times here. And there's also some more modern work that apparently can also resolve even smaller differences in terms of representations of states. So one thing that is definitely important that I take from this is it doesn't really matter that the bold response only peaks 4 seconds after the stimulus onset. If the stimulus is then shifted by 200 milliseconds and the peak happens at 4.2 seconds, then of course that there's this delay of 4 seconds that doesn't lower the temporal resolution. The question is more how reliable actually this shift in the peak of the bold response, for example, is. So that's more the question of how much certainty there is in this mapping from changes in the temporal profile of cognitive or stimulus processing processes and the peak of the bold response. Yeah? This is something to keep in mind, because it's, if you use the standard bold response model and you shift the standard bold response model by 100 milliseconds, this is in fact reflected in the signal, and hence you can also, if the signal really shows this, make claims about milliseconds processes, although the bold response extends over 4 seconds or even longer if you look at the entire profile over around almost 20 seconds. Okay? So the temporal resolution in bold is not low because the bold response needs some time to come up. Yeah? That's important. Okay, and that's also a good point for a break, because then we'll next discuss linearity properties. Are there questions right now? Good. And let's